Okay, very good evening, my dear friends. Regarding of our CPGET 2025 free online coaching, today's our eminent resource person, Dr. G.BHAGEERATH Garu, Assistant Professor, Department of Botany, Head Department of Botany, GDC Rangasaipet. Sir has clearly explained in previously about the Mendelian inheritance principles, linkage and crossing over, 3-point test cross. So in this regard, we would like to invite Dr. G. Bhagirath Garu. Please welcome and start your session, sir. Please welcome.
Thank you, sir, and welcome to online participants. Today, we learn about another topic in the genetics, that is mutations. So everybody knows, generally, the parental characters are transmitted to offsprings without any changes. But now and then, frequently we observe the offsprings which are obtained from both parents. Sometimes we identify new characteristics also. So suddenly, sometimes the new characteristics are caused by any changes or alteration in the genetic material of the organism, which is identified by some phenotypic characters which are expressed outwardly. That's why we can easily detect that change in any plant or organism or any microbe. So based on that, something is happen in the inherited material. So what is meant by mutation actually? First, we can explain about what is meant by mutation. So before going to the definition of the mutation, so the term mutation first time coined by Hugo de Vries in 1900 by his observations in Inothera lamarckiana. So evening primrose, the plant name generally called as evening primrose, the scientific name is Inothera lamarckiana. Hugo de Vries was observed that in evening primrose plant, the visually it appear in two different forms. So one is Inothera zyganthus and Inothera nanella. So both the plants are obtained in a progeny. Suddenly, why these two plants are grown in different heights? So based on that, he first time coined or postulated mutation theory. So Hugo de Vries in 1900 in Inothera lamarckiana identified two plants. So the systematic study of mutation was started in 1910 when Morgan genetically analyzed the white eye mutant of Drosophila. So once several experiments are done on different microbes like insects or bacteria or fungi, so they reveal some different characteristics in the progeny. As well as H.J. Muller also induced the mutation in Drosophila by using X-rays in 1927, and also he was awarded with the Nobel Prize in 1946. So based on that, the characteristics of mutation, how these mutations, how it manifests, or what are the characteristics of mutation. Generally, mutant alleles are recessive to their wild type or normal alleles. So that's why suddenly they appear in a particular generation. The mutant alleles are recessive to their wild type. When the absence of wild gene, these mutant allele express its character. That's why we can detect a new character suddenly in a generation. Or most mutations are harmful effects, but some mutations are beneficial also. So if in any organism or any crop plant, mutation occurs, it may be the mutation leads to benefit or loss. The spontaneous mutations are very, very low rate. For example, a farmer laying the seeds or sowing the seeds in the agriculture field, among the thousands of crop plants, one or two plants may express a new character. It is not easy to detect in the agriculture field. Such type of mutation is spontaneous mutations, which are occur in low frequency. Some genes shows high rate of mutation. Such genes are called as mutable genes. Mutable genes means in some organisms on the chromosome, these genes are undergo frequently mutations. That means high rate of mutation is present in the particular location of the chromosome which occupy few genes. Such type of genes are called mutable genes. What is that? Mutable genes. And highly mutable sites within a gene are known as hotspots. So in the biodiversity, that is different one. But in the genetics, we can call as any chromosome which containing few genes which undergo frequently mutation. Such mutable sites in that chromosome we can call as hotspots also. Mutation can occur in a tissue or cell or of an organism, or maybe in the genetic material. So these are the few characteristics of mutations. So once we can go for the definition of the mutation, then we can explain about the classification of genetics. A sudden change takes place in inherited material or genetic material of an organism, which tend to pass to the next generations. That is called as mutations. These mutations may occur in the small portion of the genetic material or sometimes larger area of the genetic material. Sometimes also we can observe the change not only in the DNA, not only in the genetic material, along with the genetic material in the chromosome also several changes takes place. So whatever the mutation, whether it is a micro mutation, macro mutation, visible mutation, so all these mutations leads to change its the characters. So that's why the mutations are broadly classified into three types. One is chromosomal mutations. Second one is gene or point mutation or factorial mutation. Sometimes we can call as micro mutations also. The third is cytoplasmic mutations. Cytoplasmic mutations means the main genetic material which is located in the nucleus only. The main material located in the nucleus only. But we can observe some mutations takes place in the cytoplasm also. Why? Because in the cytoplasm, two cell organelles are there. So the two cell organelles are called as semi-autonomous cell organelles. That is, we can call as what? So one is mitochondria, another one is chloroplast. So for example, we can take this a model typical cell of the plant cell. The main genetic material is constituted in the nucleus only. But if the cytoplasm is also containing two semi-autonomous cell organelles, this is a chloroplast which containing what DNA? ctDNA. A little amount of circular shaped DNA, double stranded DNA is also present in the chloroplast. So that is called ctDNA. In the mitochondria also we can see a little amount of double stranded circular DNA is there, that is mtDNA is there. These DNA material containing few genes which are independent one, which never depends on the main genetic material. in such genetic materials also undergo changes. It leads to variations in the next progeny or next generation. Such type of mutations are called as what? Cytoplasmic mutations. We discuss about broadly in the later classes, in the coming classes. Okay. So next I will go for the another type of mutations, that is today we'll discuss about gene or point or factorial mutations. So first one is chromosomal mutations. It is known very well. The changes occur in the chromosome structure or chromosome number. So based on that, these chromosomal mutations are classified into two types. One is numerical changes and structural changes. So structural aberrations and numerical aberrations of the chromosomes, which observed in few organisms and plants also, it leads to changes or variations in the population. So later in the next class, we discuss about the chromosomal mutations. Now we can discuss about gene or point or factorial mutations, and which are visible, biochemical, and lethal. Again, these type of mutations we can classify it into three types: visible, biochemical, and lethal. Once change occur in the small tiny part of the DNA, that is nothing but a single nucleotide. So you know very well, nucleotide containing four type of nitrogen bases: adenine, guanine, cytosine, thymine. Among the four nucleotides, one of the nucleotide is replaced with other different nucleotide, or it may be deleted, or sometimes it will be add a nucleotide in between the two nucleotides. Such type of changes takes place in the smallest area of the DNA. That's why it's called as micro mutations or factorial mutations. Sometimes we can call as gene mutations also. So this leads to change in the gene product, or it leads to change in the biochemical path or metabolism, sequential biochemical reactions in a particular metabolism also. It leads to change in the metabolic pathway, and also it changes some characters of the organisms also. Sometimes the gene mutations which occurs in the genetic material, which leads to causing the death of the organism. Such type of mutations are called as lethal mutations. అంటే ఘాతుక ఉత్పరివర్తనాలు అంటాం. ఒక జీవి మరణానికి కారణమైనటువంటి జన్యువులోని మార్పులు అన్నమాట. సో వాట్ ఎవర్ ద చేంజెస్ టేక్స్ ప్లేస్ ఇన్ ద జెనెటిక్ మెటీరియల్ which leads to causing of the death of a organism. Such type of genes are called as lethal mutations. So the biochemical mutations are nothing but a small change in the nucleotide of the DNA, replacement, addition, deletion. So these biochemical mutations again divided into two types, that is substitution mutations and frame shift mutations. So next, visible mutations. Visible mutations may be it under divided into based on the source of the mutation, how these mutations are classified. So first of all, based on the causes of mutation, how the mutation cause in the genetic material, by the influence of the human being or without any obstacle, without any influence of the human being. So generally in the evolution process, we can expect new characteristics. So these small new characteristics add in continuously generation to generation, which are manipulated, and it leads to formation of a new species also. But it takes very longer time. So first of all, we explain about spontaneous mutations, yadrichika utparivartana. So spontaneous mutations are occurs naturally without any reason or without any cause. So the rate of spontaneous mutations is very, very low and it is slow also. So that's why in a crop plants, it is not too easy to identify or detect the mutation. So the rate of spontaneous mutations is higher in eukaryotes than prokaryotes. So such type of mutations are called as spontaneous mutations. For example, a bacteria or a plant exposed to the sunlight, the natural sunlight consisting a little amount of UV light. So that's why whatever the portion or intensity of UV light penetrate into the bacteria cell, it causes a mutation. So that's why it occurs naturally without any influence of the human being. So such type of mutation is called as spontaneous mutations. And moreover, it is very low frequency and it is a very problem to detect the spontaneous mutations particularly in crop plants. Next one is induced mutations. So induced mutations are nothing but wantedly human beings improve variety in different crop plants. These crop plants are treated with different kinds of mutagens. Whatever the chemical agents or physical agents which are capable of inducing such type of mutations are known as mutagen. Mutagen is nothing but ఉత్పరివర్తన జనకాలు అంటే ఉత్పరివర్తనాలను కలుగజేసేటువంటి కారకాలను ఉత్పరివర్తన జనకాలు అంటారు. వాటిని మనం ఇంగ్లీష్లో mutagens. So by the help of mutagens we can manipulate and we can achieve some beneficial characters in the crop plants. Maybe it is a insect resistance or the high yield, or it may be a stress tolerant plant, which helps in the crop improvement program and also which helps in the mutation breeding. So example, we can use X-rays, gamma rays, UV rays causing mutations in many cereals like wheat, barley, sorghum, such type of plants. Hello? Everything is okay or any problem? Hello? Hello? Okay, this is induced mutations. So induced mutations are generally we can inculcate mutations in crop plants by using different type of physical and chemical mutagens. Example, physical mutagens are ionizing radiation and non-ionizing radiations are there. So non-ionizing radiations are nothing but gamma rays. Ionizing radiations, we can see alpha rays and beta rays, X-rays. So such type of radiations are using frequently in the laboratories to achieve maybe a beneficial character. If it may be give beneficial or sometimes harmful character also. In the same way, chemical mutagens also we are using in the laboratories. For example, mustard gas, formalin, nitric acid, and PEG, polyethylene glycol, such type of chemical components, ammonium nitrate. So these chemical compounds also used as mutations in different hybridization programs or mutation breedings. Okay. Next, we can go for based on the tissue, the mutations are original. So if mutation occurs in the somatic cells, that is called as somatic mutations. These mutations are transmits from one generation to another generation by only means of asexual reproduction. You get my point? So that means a somatic tissue helps in the any asexual structures are formed. For example, spores. Spore production comes under asexual reproduction. So that's why the character is transmitted to the next progeny. But if occurs only in the somatic cells, it may not carried by through the sexual reproduction to the next progeny. That restricted to the mutation or change that restricted to the particular generation only. It may not transmitted to next level. So that is called somatic mutation. What is meant by germinal mutations? Bija utparivartanalu. When mutation occurs in gametic cells, that is gametes or reproductive cells are known as germinal mutations. It is definitely the mutable genes are carried out through the gametes, and when gametes are united during the fertilization process, the change is continued or transmitted to the next generation and also several generations. So such type of mutation called as germinal mutations. Is it clear? So based on direction of the mutation, other one is reverse mutation. Forward mutation is nothing but a change from normal or wild type to new mutant type. Such mutation events called as forward mutations, which if the same mutant genotype changes back to the wild, for example, so a gene that is a gene is converted into A dash. It is a mutant form of the gene. So by this conversion, we can expect a new phenotypic character. That character is continued to several generations or few generations. Suddenly, after few generations, this gene again come back to the wild type. This is wild type gene. Wild type gene is capital A. A dash is what? Mutant gene. So a wild gene converted into mutant gene, that's called as forward mutation. Okay. If whatever the mutant gene which is transmitted to few generations, again it come back to the original state. That is called as reverse mutation. So that means normal or wild. So this is reverse and forward mutations also observed in the plants. Okay, so okay. Next, we'll clear the drawing. Next, we can go for the point mutations. So point mutations, again, I will explain like this.